Why we need a nucleic acid probe? We saw in cDNA library preparation that after the cDNA is made, it is cloned into a particular vector and it forms a recombinant plasmid, and we transform it into E. coli cells and we plate them. And there are many colonies. If we want to fish out a particular gene X and we want to find out which colony has gene X, we do a plate lift assay where you have transformed colonies growing on the agar surface, and we make a replica of that by placing a nitrocellulose disc on top of the colony plates, and we remove the nitrocellulose disc. We treat the nitrocellulose disc so that we can proceed for hybridization. Take the nitrocellulose disc which contains the colonies, lyse the bacteria with 0.5 normal NaOH so that the bacterial cell walls get lysed and it exposes the DNA inside, so the probes can easily bind to the DNA. Then you have to neutralize them, and you have to treat with proteinase to remove any DNA bound proteins, wash them and bake at 80 degrees Celsius so the DNA gets fixed onto the nitrocellulose membrane. Now you have to treat with P32 labeled probe. The probes are nothing but small segment of DNA which is complementary to a particular gene, for example gene X, and it is labeled with phosphorus P32, which is a radioisotope, so we can detect it using autoradiography. We will see later what autoradiography is. Then after the development of autoradiograph, we can see that this particular colony is showing some radioactivity, and thereby we can say that this is gene X for which the probe was designed. Probes can be a gene or DNA fragment, cDNA probes. They can be synthetic oligonucleotides. They can be RNA probes or riboprobes. The probes can be labeled using radioactive substances or also non-radioactive molecules. We will first see about radiolabeling. We can use a parent isotope or any element to which the DNA can be bound. And when it undergoes radioactive decay, it forms a daughter isotope and it can emit an alpha particle or a beta particle or a gamma particle. Alpha is very less penetrating, so we cannot use this. Beta is more ideal because it does not penetrate deep into our body, so it is more safer than gamma. Gamma is highly penetrating and it is highly ionizing. Since it's DNA, people always use P32 as the label. So for labeling of DNA, we can use P32. You have alpha P30, alpha phosphorus, beta phosphorus, and gamma phosphorus based on the position of the phosphorus at the 5' end of the growing of the DNA fragment. Phosphorus P32 is radioactive, and while it emits radioactivity, while it emits the beta particle, it becomes sulfur 32 and it emits electrons in the form of beta. It is called as beta emitter. And what is half-life? The time it takes for the half of the atoms of radioactive isotope to decay is called as the half-life. So the half-life for P32 is 14 days, where you can see exactly half of it has become sulfur 32. So whenever you're getting samples from outside or buying P32 from a radioactive company, you have to make sure you finish your experiments within 14 days. Otherwise, it will not be very accurate and you cannot see any specificity of the reaction. Working with radioisotope is really difficult because you have to be extremely careful, because it can ionize your bodily molecules and it is very toxic also. Before going into the radioactive lab, you should wear a personal protective equipment, a goggle, and you should also wear 2 gloves so the penetration of alpha particle is does not happen, whereas beta particle also does not penetrate when you wear two gloves. And you have to work behind a lead shield. This is the lead shield. You have to work behind the lead shield, and so that you won't get exposed. You should also wear a radiation monitor badge to make sure how much of exposure has been done from the time you started working in that lab. Labeling methods. Now, I told you that your DNA can be labeled, or fragment of DNA can be labeled, and they're called as DNA probes. And in the P32 can be attached. P32 is generally attached to the NTPs. That means it can be attached to an adenine, thymine, guanine, or cytosine. How we can attach them? You can attach through various processes. Directly, you cannot put P32 DATP and you cannot put your DNA fragment. They will not bind to each other. You will need some techniques so that you can attach the P32 labeled DNTP to the DNA fragment. That a small DNA fragment which serves as probes. You can use nick translation, primer extension, end labeling, and RNA labeling. What is nick translation? First of all, your probe has to be labeled and not your amplicon or any DNA fragment. Probe is something which you have in your hand in a tube, and you can use this for hybridization to the gene fragment which you're looking for, where you're going to fish out from a number of such genes present in the colony plates or on the agarose gel. For which you take the probe in a PCR tube, which is a small fragment of DNA, and you add DNase. DNase is an endonuclease which causes a nick, breaks the DNA strand. One strand of DNA is nicked here. And what happens now? You are adding DNTPs and also labeled DNTP. So here, for example, we can add a labeled DATP. It's not necessary that all the DNTPs should be labeled. Radioactivity can be, radioactive substances are very sensitive. It can be picked even from one molecule. So we can use either A or T or G or C, which can be labeled with P32. Apart from that, you will also add normal DNTPs and you add DNA polymerase. There's something called as nick translation, which means the DNA polymerase extends from the nick position and it creates a new strand. It replaces the old strand with a newly formed strand in which DNTPs and labeled DNTPs are added. While it is moving, since it has exonuclease activity also, then it degrades the strand, whatever it is extending. So you can see that some some nucleotides are removed while it is adding the new strand. So the final product that you get will have the new strand with the P32 labeled DNTPs added into them. This is one method by which the probes can be labeled. The other one is primer extension. When we did our experiments, we used primer extension, which is very, very simple. You take a probe of about 250 base pairs DNA. It is a double-stranded DNA. And how you get this 250 base pairs is because you take an entire gene and from which you design primers and you amplify this 250 base pair as probe. While it is amplifying, like a normal PCR, you're adding DNTPs and you can add a P32 labeled DATP. Here, except P32 labeled DATP, all other DNTPs like G, C, and T, they are all normal except ATP, which is P32 labeled at the 5' end. The 5' phosphate of the nucleoside DATP is having a p32 at the alpha position of the phosphorus. Phosphorus is replaced with the radioactive phosphorus in DATP. As a PCR is proceeding, wherever T is present, a complementary A is added by the tag polymerase, and thereby your amplicon will have a radioactive DATP added in its double-stranded DNA. Now you can use this directly in southern blotting hybridization methods or in dot blot assays, colony hybridization methods. Wherever you want, you can denature this probe into single-stranded molecule. You can also denature the DNA strand which you're looking for. You have to denature them so your probe, which is single-stranded, can bind to a single-stranded DNA. Another method is 5' end labeling of DNA. Here, the entire stretch of DNA is not labeled, but only your 5' phosphate is labeled. In cases where your sensitivity is not required that much, where you can easily pick out with just one phosphorus 32 molecule, you can use 5' end labeling of DNA. First of all, you take the probe, which is about 200 to 300 base pairs, and you have to treat the probe with alkaline phosphatase. As the name indicates, phosphatase means removal of phosphate. One phosphate group is removed from the end of DNA, that is 5' phosphate. This phosphate at the 5' end of one strand and another phosphate at the 5' end of the other strands are removed and they are replaced with OH group. Now we have to replace this OH with a radio-labeled phosphorus. So we take a kinase. So you always have to remember that phosphatase means removal of phosphate. Kinase means addition of phosphate. Here, you're replacing with a p32 labeled ATP. And alpha, beta, gamma phosphate followed by your nucleotide. So this alpha phosphate is p32 labeled. Other two phosphate gets removed because only one phosphate is added. ADP goes out and only phosphorus is remaining, which gets added here at the 5' end. So here you can see here any base, actually at the 5' end you have 3 phosphate groups, and if it is DATP, that's what we are going to add here. The alpha phosphate is p32 labeled, and the remaining 2 phosphates get removed by the kinase, and addition of only this phosphate happens or here, and the ADP molecule goes away. Sorry, it is gamma p32. This gamma p32 is added and this ADP is removed. ADP, two phosphorus means diphosphate, and remaining 1 phosphate that comes out gets attached to your DNA. Basically, you are adding a phosphorus at the 5' end of the DNA fragment with a phosphorus that is radio labeled with p32. And how you can do is, first you are adding alkaline phosphatase to remove the phosphate that is normal, and you're replacing it with the p32, gamma p32 labeled DATP. So in this figure, you can see this is alpha, beta, gamma. Now here, this part is cleaved off and it goes as ADP, adenine diphosphate. You have adenine and 2 phosphates diphosphate. And this extra phosphorus at the gamma position is p32 labeled, and that gets added at the 5' end. You can also use RNA as labels, where you can use RNA as labels, because I told you RNA is very unstable. When we are doing in situ hybridization, when we want to check the mRNA levels as such in a cell without extracting them out, what we have to do is we have to do labeling of RNA itself. So the RNA, we send them inside as the probe, which can bind to the mRNA molecules inside the cell. Now, you can easily do this by taking the RNA molecule and attaching the DATP, which can be labeled with, this phosphorus can be labeled with P32, this ATP can be labeled, and then you are adding T4 RNA ligase. Here, don't worry about this biotin or cytidine. This is an example for a non-radioactive labeling of RNA. But here, since we are talking about radioactive labeling, you can use the DATP, in which its phosphorus is radioisotope. Thereby, you can use them as probe inside the in situ hybridization processes. Basically, you can do a 5' end labeling, 3' end labeling. If you're using 5' end labeling in a DNA probe, when you're using a phosphate at the 5' end, which has to be labeled, you're using alkaline phosphatase and polynucleotide kinase. When we want to label at the 3' end, we can add terminal transferase. We know that terminal transferase is an enzyme which adds poly DT tail or poly DA, whatever. If we add more G, it adds poly G. If we add more C, it adds poly C. So if we provide only T, then it attaches the radioactively labeled DTPs to the 3' end. So you can label the 5' end as well as the 3' end. And then you can do a nick translation as we saw. First, you have to cleave it with DNase 1, and then the DNA polymerase 1 extends the nick by adding any radioisotope labeled DATPs or DNTPs. Then you can also use random hexamer, you can use PCR probe, you can use ribo probes for RNA molecules. How can you first, now you know how to label the probes. Then you know how to hybridize the probes. So how to hybridize the probes? We saw you have to take the probe, which is radio labeled, in a tube, and then you have to treat it with a nylon membrane or nitrocellulose membrane onto which the DNA fragment is bound. And you put them together in a bottle and put some buffer. Then the hybridization happens overnight at about 65 degrees Celsius. Then you have to, now it's time for detecting. Just by bringing probes and your DNA together will not suffice our experiment. What we have to do, we have to further develop, identify the hybridization, where exactly the signal is coming, which colony is positive for the gene X, we have to check. There's a method called autoradiography, Giga-Muller counter, scintillation counter, but autoradiography is mostly used for hybridization methods. What is autoradiography? You have a nylon membrane with some colonies or DNA fragments bound to the nitrocellulose membrane, which has been baked at 80 degrees Celsius. So the DNA is tightly bound to the nitrocellulose membrane. Then overnight, we have treated this nylon membrane in a bottle containing radioactive probe and the buffer. So basically, there is a long bottle and a hybridization oven which works at 65 degrees Celsius. You put this nylon membrane and fill it with the buffer, to which you also add a labeled probe. So the probe is nothing but the DNA fragment, and wherever this fragment probe can bind to the gene X, it binds here, and we have to detect them. So next day, you take this nylon membrane out of the bottle and you have to do autoradiography. So you first wash this nylon membrane with suitable buffer and keep it on a plate, a glass plate. And on top of this, you keep a X-ray sheet. exposure to the film or emulsion. So you must have seen an X-ray sheet in hospitals they use for bone X-ray. What happens is on the X-ray sheet, wherever this label radioactive isotope is there, it emits some light, and this radioactivity turns them into different color. Basically, this X-ray film is made up of silver bromide. The X-ray sheet that you see, which is in blue color, basically it will be silver. It is coated with silver bromide. And wherever this radioactivity happens, when wherever the probe emits the radioactivity, then it gets converted to silver ions. Now you have to develop this X-ray film using certain solutions, which you see in places on photo in studios, where what happens is you treat them with solution and you develop the negative of the X-ray sheet. Now on the X-ray sheet, you will see dark color patches, and wherever this radioactivity has happened, the remaining place will be blue in color, and where your radioactivity has happened, that will be black in color. Here you can see this is the autoradiograph of the colonies. Wherever the colonies are positive, you have treated the probe with the nylon membrane. Wherever there is positive, wherever the probe has bound to the DNA in the colonies, there you see a dark patch or dark blotch. This is called as dot blot. Dot blot. Where directly you can hybridize the colonies with probes. And these colonies are positive colonies in autoradiography, which you can detect. So in autoradiography, how you do is you take the nylon membrane to which the probe has been bound and keep them on the X-ray sheet. The X-ray sheet is made up of silver bromide. Wherever this radioisotope hits the screen, there the silver bromide gets converted to silver ions. And these silver ions, when we develop, they form a black color blotch on the X-ray sheet, and the remaining areas of the silver bromide gets washed away and they are blue in color. So whenever you at home, if you have, you can take an X-ray sheet and see the remaining areas of the bone. Wherever your bone is scanned, your bone is seen in white color, and the remaining areas will be either black or blue in color. This is the most commonly used method for detecting the hybridization of the probes onto your DNA molecules. Gigamuller counter. This you must have seen in Kalpakam or any atomic stations. People used to carry some machines with them to detect the presence of autoradiography or the radioactivity. This is very simple, but this is not very specific like your autoradiography. This is a meter which has a tube which is filled with argon gas, and it has an electrode, cathode, and the anode. It has the anode and the cathode. And it has a thin window in which penetration of the radiation can occur. For example, on your X-ray sheet or on your nitrocellulose membrane, if there is some radioactivity, then that radioactivity, the radiation penetrates through this window. And what happens here is when radiation collides with gas atoms, when the radiation alpha, beta, gamma, alpha will not penetrate, beta, gamma, when it penetrates, then it collides with gas, argon gas, and it ejects some electrons and the ions are formed. Then electricity is produced in proportion to the amount of ionizing radiation. The more the ions are formed inside this tube, the more the deflection of this needle, here there is a needle, the more the deflection towards the right side, because more current is produced because of this ionizing radiations. which ionizes the gas present inside the argon gas present inside these tubes. Let's not go more into the chemistry of this. This is basically to detect the radiation present on any place, and it's not very specific or sensitive as compared to your autoradiography. Moving on to non-radioactive labels. This is very simple and it does not require any dedicated rooms, does not require any personal protective equipment, so staff monitoring it, and also it's not very sensitive as compared to your radioactive probes. As the name indicates, here we are going to attach the probe with a non-radioactive substance or molecule. Probe is nothing but a DNA molecule or a DNA fragment which is complementary to the target sequence or gene X. So you should always imagine in your mind that when we are talking about probe labeling or hybridization, your protein or RNA or DNA is always always fixed onto a nitrocellulose membrane. And to this membrane, we are going to treat the probe in a buffer. So everything is in a solution. After you fix the DNA onto the nitrocellulose membrane, you add the probe in a buffer of about 100 ml and put them in a bottle and leave it for hybridization. Whether it is radioactive or non-radioactive, you follow the same methodology. So when we are talking or discussing about probe hybridization, you always should imagine that everything happens on the nitrocellulose membrane. Here, the probes can be a fluorophore. The molecules to which the probes are attached can be a fluorophore or haptens. Haptens means a molecule which has antigenicity to which antibodies can bind. So it can elicit immune response in a body. That means it will be able to generate an antibody against it. For example, biotin digoxygenin. Here, this is the target gene X, and this probe is complementary to the particular gene. This probe is attached to a fluorophore. Thereby, directly you can check on a fluorimeter. Or you can attach this probe to a hapten. Here it is biotin. Biotin is a vitamin H which has high affinity to streptavidin molecule, and this streptavidin can be attached to a fluorophore or it can be attached to an enzyme. Now we have to take the blot and put it on a solution containing the substrate for the enzyme. Now wherever this DNA is attached to the nitrocellulose membrane, there alone you will see some color based upon the substrate and the enzyme we use. So if we use X-gal, you will see a blue color band when the substrate is X-gal and the enzyme is LAGZ. When the enzyme is LAGZ, the substrate we use is X-gal, and you see a blue color band on the nitrocellulose membrane. You can also use digoxygenin to which antibodies can bind. The antibody can be a secondary antibody to which a fluorophore can be attached. Thereby the fluorescence can be detected directly, or a reporter enzyme like here can be attached. So now you treat the entire nitrocellulose membrane in a substrate solution. You pour the substrate solution on this membrane, and wherever the enzyme is present, there alone you will see a blue or a different color band. So nitrocellulose membrane will show up colored spots or colored bands, which means at that point you have the particular DNA or RNA or protein. That's how you hybridize with the probes. Probes can also be used for Western blotting. Probes here are nothing but primary antibodies to which your proteins can be bound. This primary antibody is developed against the protein that you're working with. Then you add the secondary antibody, which can bind to the primary antibody. The secondary antibody is labeled with a certain enzyme. Here, as you can see, the protein on the nitrocellulose membrane, or protein on the nitrocellulose membrane, it's attached to the primary antibody. The primary antibody can attach to secondary antibody, which is bound to an enzyme called as horseradish peroxidase. Horseradish peroxidase. When we add substrate, this horseradish peroxidase converts the substrate to a colored product. Thereby you can see colored bands on the nitrocellulose membrane in a Western blot. We already discussed about the disadvantages of radioactive labeling. Radioisotopes are difficult to handle and they are expensive to dispose of. We need to pay to some companies for them to take our waste away. If there are few counts in hybrid detection, then autoradiography will take long time. For example, if your gene X is of very less copy number, then it might take a longer time for the autoradiography to happen. The signal will be very less, so detection will be very late. And radioisotopes have a short half-life, and therefore experiments should be completed really fast. I told you it's 14 days for P32, so you have to finish your plan, your experiments first, get your blots ready, then you can order for P32 from the company. Applications, you can do a southern blot analysis to detect the DNA molecules, northern blot to detect the RNA molecules, dot blot to detect colonies containing the suitable genes, and you can do a plaque blots when you're using phages, bacteriophages, and you can also do in situ hybridization directly on clinical specimens in live cells. Thank you very much.